I'm not going to become bashful and say this, that, or the other. I'm just going to sip on my brown liquor here. So look at these pictures. Look at the title of my video. The video is called DNA, Genetics, and Zygosity. And I just wanted to explain how deoxyribonucleic acid is the vehicle for genetics, and that those same genetics are expressed through zygosity. It would be far too much for me to do that. You know what? I'm looking at 17 minutes and 20 seconds of 20-second frames. And I'm thinking about shortening it down to where it'll be back down to its normal 6 minutes and some change seconds. Really. But I'm going to read this stuff. And it just depends on how I feel, how I'm going to go about all of it. It's not that this is a thankless effort. It's just that I assume everybody already knows this shit. The way that they fucking act. Right on? Because if I kept it all to myself, it wouldn't make a difference in my life. Can you dig that? You got motherfuckers who come around here, talk the most absurd nonsense. Then wonder why a guy like me just sits back in the cuts. Because I could watch you destroy yourself and not feel no type of way about it. Almost feel good knowing that that was somebody that I didn't even care for in the first place. For my people, I'm not talking to you. For those other things that come around here, I'm talking to them. Because they come around, you see, seeking to scrounge up whatever little pieces of information that they can and then use it as it's some type of a weapon against what it is that I already stand for. I stand for righteousness with my people. The only reason why I'm telling you guys about genetics, DNA, and zygosity, because I've been stressing this shit since I first talked about it. Do you know what this has to do? What it goes back to, everybody? It goes back to blood mixing. Right. Why would he do all this just to talk about blood mixing? I know you're wondering, right? What do you think crushed Israel? Huh? What do you think did him in? Genetic constitution is huge. Can you dig it? Genetic constitution is huge. It's epic. So let's go to Google first. No. Let's go to twins.org.au. We're going to read about the different types of twins. And keep this in mind about the stuff that we've already read through in the Bible. Listen. Twins, triplets, quadruplets, whatever. It's all the same. Okay? Apply these to what you know. How to determine zygosity is what it says here. For twins, zygosity refers to the degree of genetic similarity within each pair. There are two different types of twins. Dizygotic, fraternal twins, and there are monozygotic, identical twins. We're going to read about the DZ, or the dizygotic, first. Those twins occur when two eggs are released at a single ovulation and are fertilized by two different sperm. Those two different fertilized eggs then implant independently in the uterus. DZ twins share around 50% of their genes, which is the same type of genetic relationship as non-twin siblings, hence the term fraternal. And approximately half are the same sex and half are different sex, so it's 50-50. Each has its own placenta, which is continuous with the chorion or outer membrane, with rare exceptions. Each has its genetically each is as genetically similar as any same-sex or different sex siblings. Monozygotic, MZ, identical twins, develop when one egg is fertilized by a single sperm, and during the first 2 weeks after conception, the developing embryo splits into two. As a result, two genetically identical babies develop. Approximately 1/3 of MZ babies have separate placentas. So 1/3 of these identical babies have separate placentas. Approximately 2/3 of MZ babies share a single placenta, despite maintaining their own inner sac, amnion, or umbilical cord, and share of the placenta mass. All MZ twins are the same sex with rare exceptions. All are genetically identical or almost 100% identical. A zygosity diagram can be downloaded here. I'll see if it's really that good and then I'll try to use it. But check this out. Dizygotic twins would be who you know as, what's his name? Jacob and Esau. Those are dizygotic twins. Monozygotic triplets that we know of in the Bible are known as Abraham, Nahor, and Haran, or Nakor and Aran, and Sem, Kam, and Japhet, or Shem, Ham, and Japhet. I'm going to keep it pushing. Zygosity. It's a noun. It refers to genetics. It is the characterization of an individual's heredity traits and a term of gene pairing in the zygote from which it is developed. So, once again, homozygous, heterozygous. What you guys just heard about as far as dizygosity and monozygosity is almost the same as homozygosity and heterozygosity. Can you dig it? Heterozygous twins. Homozygous twins. Right. The characterization of twinning and multiple births in a term of the combination of alleles for particular hereditary traits. That is the second term of zygotes or zygosity. I need y'all to understand this. This is bigger than me. Okay? This is much bigger than me. And I have nothing to win from this. Let's read about the word heterogametic real quickly. Denoting the sex that has sex chromosomes that differ in morphology. Hold on. When it speaks on morphology, it's pretty much talking about the way that genes develop. So when the little baby is sitting inside of the egg, the sperm fertilizes the egg and is chilling in the placenta. It's talking about what sex it will take on, and it says pretty much that heterogametic species, or the species that is heterogametic, or the sexes that is heterogametic, right on. In mammals, it would be the males, and in birds in particular, it is the female. Why this is so important is because, once again now, this is the species or the sex that will give off to the offspring its genetic coding as to it will be a male or it will be a female. Why this is important is because, once again, we're talking about the twins right now and these guys, right on. So we're going to talk about how the fact that it works 50-50 in a pairing of two different people, but it doesn't work like that with very rare exceptions. Very rare exceptions. And I've seen the rare exceptions with white people. I know a set of white triplets, 2 chicks and 1 dude. So I know what it's talking about when the rare exceptions. I've seen it. Anyway, that's nothing. And I went to school with them. Y'all got to understand how it works. And really, really dig it. Now, we're going to talk about the next part of this stuff. Because the next part of this would be the Nuremberg history section. And we're going to read the identifying of DNA. Here we go. The first step was to identify DNA. Oh, excuse me, I lied. Let's go back. We're at history.nih.gov. Now, let's carry on. The first step was to identify DNA. And the reason why I'm saying that is because it's picking up off where people had just discovered that Gregor Mendel had pretty much discovered or was the father of genetics. Right on? So they're picking up from there and they're going from there and they're saying, okay, well, what can we do with this information? And they say to themselves, well, what was the first step? To identify DNA as the carrier of genetic information. Stop right there. So they're not trying to disprove genetics. They're trying to prove genetics. In fact, they're trying to prove genes are so true that they're trying to show that DNA even that genetics have a vehicle, and its name is deoxyribonucleic acid. It was discovered by Oswald Avery. He was working at the Rockefeller Institute, in New York, right? And pretty much he discovered that the pneumococcus organisms or disease-causing bacteria, right on, were neither a protein or lipids. In 1944, himself, including Colin McLeod and Macklin McCarty, they published a paper that suggested that DNA was responsible for transferring genetic information. Hold on, and this idea was not easily accepted. Now, the reason why this is not easily accepted is because they're just trying to accept that genetics are genetics, genes are genes, right? And they work themselves out. But the reason why they needed for people to understand this is that because DNA can be healthy, it can be unhealthy, right on? You can have damaged genetic material. However, this is obviously, we're talking about now, not then. These guys are just trying to show people that this stuff was real. So, and it says here, after reading Avery and McLeod's and McCarty's paper, many scientists changed the focus of their research to further investigate nucleic acids. One of the most successful, Erwin Chargaff, found a clue when he discovered that makeup of DNA differs from one species to another. So DNA is not the same from one thing to the next. So the fact that they could use E. coli, right on, as the base understanding for DNA is crazy. But the reason why they were able to use E. coli was because they had they had genetic material samples from humans. However, let's go keep it pushing. He also studied the ratio of bases in DNA of different species and concluded that the two bases, adenine A and thymine T, appeared in relatively equal amounts, as did guanine G and cytosine C. This helped pave the way for discoveries to come about the shape of DNA. Now, let me just break something down to y'all. The reason why they say that the shape of DNA is important is because you have to understand the breaking off of different genetic materials, things that they call pendants, okay? And these pendants change the actual course and the identity of genetic material itself. Depending on what those pendants are, pretty much make up what that genetic material is. Now, what you are made up of, you can only put that out. Carrying on. In the early 1950s, then scientists knew that DNA was genetic material. So understand, DNA is genetic material. It is not something that is not of. It is exactly that. And it was formed of sugars, phosphate groups, and equally matched bases that thymine, cytosine, what's the other one? Uh, Guanine and adenine. That's what it's talking about. Right on. They tried to pull the okie doke with the words, but it's the same stuff. James Watson and Francis Crick worked together at Cambridge University in England and assimilated this information along with the help of Maurice Wilkins, an expert X-ray crystallography images and expert X-ray crystallography images prepared by Rosalind Franklin, both of King's Colleges in London. In 1953, Watson and Crick came up with their historic model of the shape of DNA, the double helix. One moment, here we go. As Watson and Crick discovered, DNA is in the shape of a double helix, with the outer strands made up of phosphate and sugar combination. The base pairs linked to the forms of inner ladder-like rungs that hold the outside strands together. Identifying the shape of DNA was a major breakthrough in genetic research, for which Watson and Crick and Wilkins won the Nobel Prize for Physiology or Medicine in 1962. But how did the information of DNA get translated into proteins? What was nature's genetic code? Now let me just stop right there. That's the end of this article. Now, if you guys know anything about the double helix and the guys that discovered it, you know that they did LSD to discover the double helix. Right on, they did drugs. All right, they didn't just fucking discover it. However, this video is to supplement and give information to the things that people don't talk about. Can't stress it enough, y'all. I could hold it all to myself, it wouldn't make a difference. I'm praying that this does help somebody, some way, shape, or form, though. Thank you for your time.